Hello, everyone, and welcome to today's live broadcast, using genome-wide sequencing to identify gene-gene interaction, presented by Dr. Christopher Amos, Associate Director of Population Scientists, Professor of Community and Family Medicine at the Geisel School of Medicine, Norris Cotton Cancer Center, Dartmouth College. I am Alexis Carlos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before I begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Christopher Amos. I will now turn the presentation over to him.
Thank you for the introduction. It's a great pleasure to be presenting to you today. So I've slightly changed the title, although I will be covering the topics that were discussed. So the title is Toward Precision Medicine for Cancer Prevention and Early Detection. So what is precision medicine and why do we use this term? Since clinicians are always trying to match their patient characteristics to the most relevant treatment, basically, you know, we're undergoing a revolution in genomics and other high throughput profiling methods. That's because the technologies have evolved very rapidly and the costs for completely querying a person's genome and attributes of the disease have fallen precipitously, making it possible for us to characterize what's happening in a disease process very well. And that's allowing us to provide stunning improvements in some cases to treatment. And so we're going to talk about how those changes can impact our understanding of early stages in cancer development. And so with precision medicine, we can evaluate individual specific mutations or polymorphisms and changes that are occurring in expression of RNA, and so better tailor the disease to the specific characteristics of the of what's happening with it. So in this slide, we're showing a diagram of the general view of cancer development, in which there are early changes that occur in the tumor, and subsequently additional mutations can occur, and those lead to heterogeneity in the tumor and may adversely affect its behavior, making it harder to treat in many cases because there are different branches of the cancer, different components of it that may respond differently to treatment. However, in many cases, those early stages may determine a treatment option, and in particular, germline changes are relatively common. There's several recent studies coming out of, for instance, Sloan Kettering, showing between 15 and 20% of patients that have been routinely screened for cancer having a germline variant. And in the slide, this is showing you results from the TCGA for colon cancer, and they're showing that colon cancers show a diversity of spectra with respect to the prevalence of mutations that occur. There is a set of mutations with very high mutation load. These are individuals have often germline mutations in one of the DNA mismatch repair genes that make them more susceptible to developing many, many more mutations. And on this next slide, we're showing you results of a study, a landmark study that showed that those patients, overall, colon cancer did not respond to pembrolizumab, which is a PD-1 inhibitor. It's also known as Keytruda in the U.S. So overall, colon cancer patients didn't respond much better than on the treatment than was expected. But it turned out that if you had a mismatch repair deficient tumor, there was an excellent response, whereas if you were not carrying a mismatch repair defect, there was no improvement. So this is showing that germline variants can have a profound impact on response to therapy. And this kind of finding extends to several other kinds of cancers. For instance, if you have an inherited mutation in BRCA1 or 2, and you have ovarian cancer, you respond much better to a PARP inhibitor. And so those kinds of germline variants are determining the way in which treatment should be applied. So the rest of I mostly work on lung cancer. So the rest of my talk is talking about how best to understand early stages of lung cancer. And the reason that that's interesting is, first of all, lung cancer is often associated with a bad outcome, and that's because most cases are detected at a late stage. We do know a lot about etiology of lung cancer. Something like 85% of the attributable risk for developing lung cancer comes from cigarette smoking, and there are other occupational exposures that can increase risk, but also we know that the genetic milieu may support or protect from getting lung cancer. So, early stage lung cancer has a much better prognosis than late stage cancer, especially true from lung cancer and many others. One of the more recent findings was that this large trial, the National Lung Screening Trial, showed 20% reduction in lung cancer among those individuals who had low-dose spiral CT, and the mortality, overall mortality, was 7% lower. So this is relatively unusual case in which screening and early detection actually reduced overall mortality, not just the cause-specific component. And also there is a dramatic stage shift so that more cases were detected earlier, at earlier stages. So that means that we will be detecting more early stage lung cancer as the screening becomes more prevalent. The problem is the cost is quite dramatic, and the estimates are that the cost of Medicare would be $1.3 to $2 billion a year. So right now, screening is restricted to individuals who have extensive history of smoking, and in some cases, family history can enter into the screening guidelines, but this does leave quite a large population that are at risk for getting lung cancer not being screened. So it's nice to have this option, but it's somewhat incomplete and expensive. As I mentioned, we do know that smoking is a major driver for lung cancer. This slide is showing what happens among those people who keep smoking. So risk keeps accumulating. If you quit smoking, risk levels off at the point at which you have quit. But it still increases, sort of paralleling the rate of increase associated with age. So it's not like cardiovascular disease where risk goes back to normal. It always, your risk always stays elevated, but it just parallels where it was at the point at which you quit. So another kind of comment here in terms of behavior, we know smoking is bad, it increases risk. Unfortunately, with e-cigarettes available, we have a new sort of population of teenagers who are becoming dependent on nicotine, and there's quite a bit of crossover between e-cigarettes and cigarettes. So there's still a population at risk, and also the U.S. and other countries, smoking continues to be a problem. So in lung cancer control, we'd like to minimize risk. So to do that, understanding how genetic and environmental factors jointly influence cancer risk is important. And tobacco smoke has many constituents, so genetic studies may provide some insights into the mechanisms by which smoking behavior is maintained, and also potentially how those harmful components in cigarettes are influencing risk. Also, we can use genetic information to shed light on other factors, host-specific factors that influence cancer risk. So I'll get to that at the end. And then we may identify subsets of people who benefit the most, and potentially some people that would appear to be at higher risk who won't benefit as much. So in the background, continuing, there are some high-risk syndromes that predispose for greatly elevated risks. So people with an inherited mutation of retinoblastoma have about a 15-fold higher risk of getting small cell lung cancer. And we know that small cell lung cancers often have retinoblastoma mutations. So those two are tightly connected. People with P53 mutations, they're quite uncommon, but their risks for getting lung cancer are extremely high, and they increase if they smoke. And then there is a homozygous mutation of EGFR, which is rare, that is associated with lung cancer. Groups done modeling in families and showed that there's evidence that in families there's a dominant genetic factor that can be inherited. So this is a slide just showing you if you have an inherited P53 mutation, that's called Remini syndrome, and it's characterized by high risks for getting breast cancer in women and multiple other cancers like leukemia and sarcoma. But shown here in the circles are cases of lung cancer. Also head and neck cancers occur. So we did a study earlier this in the 2003 that showed what the high risk of getting lung cancer is among the carriers of P53 mutations. So as I mentioned earlier, evidence for dominant inheritance of susceptibility for developing lung cancer. And so in order to identify those genetic factors, we organized a consortium that collects samples from multiple sites in order to identify those strongly acting genetic factors. The problem with studying lung cancer in this kind of family design is that, as I mentioned earlier, it's often lethal fairly quickly, and so it can be quite difficult to get samples from multiple relatives. You can get the sample from the index case at the time when that person is diagnosed with lung cancer, but trying to collect data from the relatives is more challenging. One way to do that is to collect samples from other tissues that have been collected, for instance, tonsillectomies, or if they had lymph nodes resected at the time of a surgery. Another way to do that is to reconstruct the genotypes of affected people from the offspring. So if you have one parent, let's say the person without lung cancer that was married to the case, and children, then you can reconstruct the genotype of that parent. And if you have multiple relatives and you're missing some, you can still reconstruct with a high probability. So this requires, sort of an extensive network to manage the collection, the genotyping, and the organization of data. But we've been doing that. And so this is showing you a kind of pedigree. So looking at pedigrees, the squares are males and circles are females, and the affected people are shaded. Kind of structure, you see lots of lung cancers and some head and neck cancers, and we really didn't see other cancers showing up excessively. So it's really just a lung and head and neck kind of cancer structure. So when we analyzed these data, we found a region on chromosome 6q that was supporting evidence for a genetic factor. That evidence is measured by something we call a LOD score, and LOD scores over 3 are considered very strong evidence. In this diagram, we're showing that the evidence for an effect is really restricted to those families that have 3 or more affected persons in the pedigree, and really it's the 4 or more in a pedigree that is driving the evidence. So the problem with a link analysis is it does identify regions, but they can be quite broad. So trying to identify the specific genetic factor that is driving this evidence for genetic susceptibility is challenging. But we did identify a variant that's increased, that's associated with the increased risk in some families. So the way we did that is we selected a family and we evaluated the relatives in the family that had lung cancer, and we compared those cases to other individuals who didn't have lung cancer. And by doing that, comparing the cases from sequencing with lung cancer to the controls, so relatives that didn't have cancer, we were able to, and looking in that region, we're able to identify a few variants that were co-inherited with the disease and not with the unaffected people. And in particular, there's a variant of the PARC and PARC2 that was strongly associated. And this slide's just showing you this variant is in a ring finger domain, and it has a functional impact. So it's a pretty strong candidate, and it was found in several other families, so at least explains some of the variability, but probably not all of the risk associated with this region. So we're continuing to do the studies on high-risk families, and the advantage of families for this kind of study is that we, again, can have multiple affected individuals in the family and compare them to controls in the family. The best kind of control would be someone who marries into the family and not someone who's an offspring who could be at risk. But anyway, the other individuals in the family are good. They match the cases in terms of the environmental factors and probably ethnic factors to a large degree, and so that those make it easier to sort out what a causal variant is in a family. If you're just sequencing random individuals, there's a tremendous amount of information that you obtain. If you do a whole genome sequencing, you'll end up with about 3 million variants per person. And without some strong way of filtering that information, becomes challenging to assign causality to the variants that you identify. This kind of analysis can be done if you have large populations of cases and controls. Another caveat with that kind of design is that you need to be sure that the control sequencing and the case sequencing is done in the same way. Sometimes people choose cases and sequence them in families and then compare the sequences to some control they might get off the internet. The problem with that is if the sequencing is done in a different manner, you'll get different variants being identified. And so while it's not always necessary to have the controls match perfectly to cases in terms of their exposures, I mean, that is the best. Definitely the most important thing is to make sure that the sequencing process is the same between the case and control groups. So on this slide, we're showing you sort of the range of variation that exists in population. So what I have been talking about until now was high-risk families. In those cases, we have multiple relatives affected with a cancer or some disease, and we can use the occurrence of the disease in the family to help us identify the variant, but that requires that there's a high penetrance, there's a high probability that a person will get affected given that they have a mutation that predisposes to the disease. Those kinds of high-risk variants are generally not very common in the population because they're selected against. On the other hand, there have been many studies trying to identify low-risk variants that are quite common. These can have substantial impact at a population level. There are many of these. For each individual, they don't have much effect, but when they're aggregated, they can identify some individuals that have high risk because they have multiple variants. They're also very useful for understanding the biology of the disease development, so they do play an important role. In the middle, there's the sort of medium effect, and relatively uncommon. Those are actually quite hard to identify, and that kind of design would be select cases with lots of relatives and trying to sequence the cases rather than trying to collect the whole family. That because in that case you need relatively large sample sizes, and the families are going to be less informative. But so for this talk, I'm switching to the sort of low effect common variants for the rest of what I'll be talking about. So in that case, what we do is a kind of case control study. So we just collect a bunch of cases, usually unrelated to anyone else, and then we collect controls, people that don't have the disease, that had a similar exposure pattern. We often require that they're from the same ethnic population. The reason we do that is because different ethnic groups have different alleles, and if we don't control in some way for ethnicity, then that becomes a confounder, and we may end up identifying lots of variants that identify the ethnicity if the case and control populations vary a little bit with respect to what their ethnic background is. So we either have to control for ethnicity in the analysis or restrict to more homogeneous ethnic populations. And then if we want to combine the data, then we would do the analyses within ethnicity and then do a comparative analysis or a meta-analysis across ethnicities. One thing about these kinds of studies is, as I mentioned, the variants that we're seeking to identify are common, so that's a good thing, but they tend to have low effect sizes, and therefore we have to have very large sample sizes to collect enough individuals to be able to detect those low effect sizes. And so this is a study that we've been involved in. We, at this point, when the slide was made, we had about 20,000 cases and about 30,000 controls. And the analysis showed a very striking association with a region on chromosome 15 that contains nicotinic acetylcholine receptor variants. At the time when these were identified, they were kind of the ugly ducklings of nicotinic acetylcholine receptors because they're expressed at relatively low levels in the brain and expressed at relatively higher levels in other tissues. So they weren't really the receptors that were being studied by most neuroscientists. More of interest would be like chRNA4 and chRNA7. But anyway, we identified these variants, and so then the hunt was on to try and figure out what they're doing. And at the same time, the same region was being identified as playing the most important role for tobacco smoke use. Again, that was surprising. Mechanisms weren't really known for how those receptors would be playing a role in tobacco. So our resource was able to show that these there's a couple variants that are very important and play a big role. One of them has been called Mr. Big by Laura Byroot. That's RS169699. This is a variant that affects signal transduction through chRNA5, and it was found to play a role in a number of cigarettes smoked per day, and it has a very important role in smoking cessation. It seems people with this variant have more of an impact, a negative impact when they try to quit. And there's another variant in the region that affects expression of chRNA5. So the two variants co-occur on chromosomes, and when they do that, they form a haplotype that is a higher risk haplotype for higher amounts of smoking and more difficulty in quitting. So Laura's group, this is work by Li-Shen Chen, we're finding that when you have, so they took data from several existing studies that compare the ability of individuals to quit smoking if they had either placebo treatment or they got a pharmacological intervention. They could either get bupropion or varenicline, and if they had the high risk haplotypes, if they had two copies of those, the high risk genetic factors, then the impact of treatment was dramatically better response. On the other hand, if they didn't have the variants, then there was no impact of pharmacological intervention. So this was results from several different trials, but they were post hoc analyses, and therefore their group, along with Karen Lerman's group in Philadelphia, are involved in trials to understand if allocating patients based on their genotypes will improve the ability to quit more effectively. The implication from this study is that you should probably allocate people to treatment according to their genotypes, because if they don't have the risk genotypes, then they won't benefit from this kind of pharmacological intervention. So we also have now more recently designed a very large study And what we did is we brought together all the groups studying common cancers and designed an array that has about 500,000 markers that pass all the quality control steps. And that includes a set of markers that are like roadmaps across the whole genome to identify variability, as well as targets. So we have pharmacogenetic components, and we also have some regions that we were particularly interested in, and also variants that will inform about risk factors like BMI, age at menarche, and so these were all put onto this array. And because we worked together, we were able to reduce the cost of the array to about $40 per person that was analyzed. And then the array was configured that it could be changed if other groups wanted to join in. They would get the 600,000 B types, but that turns into about 500,000 genotypes. Anyway, all other groups could join in, and in all, about probably 500,000 or so people have been genotyped on these arrays. This just shows you the different consortia that formed the array in the beginning, and those acronyms are for the different groups like lung and colon and so forth. And so, as I mentioned earlier, in order to make an analysis that is free from confounding by ethnicity, you have to estimate the ethnic background, and that becomes very challenging with such a huge study. We developed a method that will that's effective to apply across a large study, and what it does is it uses the fact that in this figure, you can see on the lower right-hand side, those are the genotypes projected onto principal components of the European descent individuals. On the left-hand side, we have African descent, and on the top of the triangle are Asian descent populations. So what we do is we take each individual and project them to the nearest position on the triangle, or if they're in the center, to the different positions on the triangle. And so by doing that, we can estimate the ethnic background of each. Typically, for a European-only kind of analysis, you would restrict to individuals who are estimated to be about 80% European. For African American and Asians, the sort of profiles may vary a little bit, but that's the way that we typically do the analysis, to try and restrict to better, more homogeneous subsets so that we don't get confounding by ethnic background. And so this shows you some results for lung cancer when we apply that array to European-only populations. And basically the take-home from this slide is that adenocarcinoma and squamous carcinomas look very, very different in terms of their genetic profiles. There is similarity on that chromosome 15 nicotinic acetylcholine receptor region, but otherwise also some similarity on chromosome 5, where the H-TERT is a gene that relates to telomere function. Otherwise, they look quite different. And in that study, it was published in Nature Genetics in 2017, we went further to try to understand some of the new variants that we identified. The way we did that is we connected on the x-axis, we're showing you the results from EQTL analyses. So what in a region. So we picked our most associated SNP, and then we looked in about a 200 kilobase region around that SNP. And we received data from GTEx, which is a resource in which people have contributed samples, and there is array data, like genotype array data, and also expression array data. So for each SNP in a region, you can see how much of the variability in that SNP is in that expression profile is being explained by the SNP. And you can also, on the y-axis, we're showing a z-score for association of the SNP with lung cancer risk. And by plotting these two things against each other, we can find that in some of these regions, the SNPs, the most significantly associated with lung cancer SNPs, are also very strongly associated with expression. So for these 4 different regions, we're finding that the SNP that we identified is affecting, most likely affecting lung cancer risk by affecting the expression levels. So that actually makes the hunt for the mechanism for how the SNP's affecting disease easier in these cases. There are many other ways in which SNPs can affect or can be tagging variation that affects a disease, and often that's by very subtle impacts on the expression pattern. So sometimes it's quite difficult, but in these cases, I think it's going to be easier. So for lung cancer, why some people are protected. So we know people that have smoked a lot for years and they don't get lung cancer. So why is that? Turns out that's not a very easy question to answer. All interaction studies need much larger sample sizes, and that was a rationale for doing this study, was to assemble enough people that we could do gene environment studies. And so we recently published a paper where we did a rather intricate analysis in which we started with a case-only approach in which we compare the genotypes of cases that according to smoking behavior. And so that allows for an interaction analysis, which is more powerful, it turns out, than a case control design. The problem with a case-only analysis is it's also sensitive to any kind of substructure or ethnic substructure in particular in your population. So we followed that up with a case control design, which is not as sensitive to that. And then we did replication, and by doing that, and this is sort of showing the statistical analysis. So first we have a case-only design, then we have a case control with an interaction component. By doing that, we found 3 different regions, and this is just showing you the regional plots of the association of the SNPs with the interaction term. In this kind of complicated slide, I'm showing you that actually these interactions are ones in which there's a protective effect. So in these cases, having both smoking and the genetic factor combined turn out to be less severe of an effect than you would predict from having either smoking or the genotype. So this is ends up finding some of those protective factors. I think further work would be helpful. We use an extremely conservative approach, so we're quite confident these are real, but we could also do more with by not being so restrictive in the analysis. So now we turn to other questions, like how can we improve lung cancer detection? So we know, like, epidemiological models do actually pretty well. They can predict it between 75, some cases up to 80% of the risk by allowing for sort of polynomial effects of smoking on risk. Genetic markers can improve the risk by 4 to 5%. But what about other factors like BMI, telomere length, and can we identify some other biomarkers that would improve the risk prediction so that we can even further restrict the analyses, the screening behaviors to those that would benefit the most? So one ongoing question in lung cancer research is whether BMI is a risk factor. So if you just select cases of lung cancer and study them, you'll find that they have lower BMI than the general population, but that could be problematic. We know that if they're sick before, like if, it takes several years at least to develop a clinically apparent case of lung cancer, and during that time, the patients may be losing weight due to the presence of the cancer. Also, if they smoke, they're more likely to eat less. Smoking is a bit of an appetite suppressant. So there's also some association that high BMI increases lung cancer risk among non-smokers. So it's kind of not the best when you have BMI associated with increased risk in non-smokers and decreased risk in the smokers. So, and this is just showing you, like if you analysis naively, you will find that lower BMI is a risk factor. Higher BMI is protective for lung cancer when you do the analysis naively. If you try to do a prospective study, so you collect BMI several years before the cancer becomes apparent, there's less of an effect, but still lower BMI is somewhat of a risk factor. So there is a technique called Mendelian Randomization, and the idea here is that you use the genetic factors to exclude the effects that may occur due to confounding. So in this case, smoking is a confounder for the effect of BMI on cancer, or reverse causality. And in that case, reverse causality is when the presence of lung cancer is affecting BMI. So what's done is you first regress the genotypes on the phenotypes, so that's G to P, and you get an estimate of the relationship between, in this case, BMI and genetic factors. And you also assess the risk, the association between the genotypes and the disease outcome. And so then by forming the ratio of those two regressions, so the regression of the disease on the genotype and the regression of the disease on the phenotype, then you can get an estimate of the effect of the phenotype, the BMI, on the disease outcome. So as an example, we can look at body mass index. So the observational estimate is that having a higher BMI is protective for getting lung cancer. And then in this earlier study, what was found with 28 SNPs is predicting the BMI, that actually it looks like BMI might be become a bit of a risk factor. So this kind of analysis has been recently updated. So in this analysis, they first checked that they had a good set of genetic predictors for BMI, and they show in the top part of this figure that in both cases and controls, the set of SNPs they're using to predict BMI really do work. And they also show that those same genetic factors don't predict smoking behavior. So that means that you're not getting some kind of confounding from the SNP predictors on smoking. So that was a very good quality step that they did. Then when they applied this analysis to that data that I showed earlier from our consortium, they see some evidence for an effect of BMI on risk for lung cancer, but actually further analysis shows that that association is restricted just to those patients that develop squamous or small cell cancer, and for adenocarcinoma, it might be a little bit protective. So this is where this kind of association can improve our ability to estimate the effect of a host-specific factor, in this case BMI, on lung cancer risk. And so again, it's suggesting that increased BMI is actually the risk factor for getting lung cancer. So another question you might ask, what about telomere? So telomere length shortens with age. Cancers can develop among individuals with short telomeres. We do know that exists. And there were several papers that showed an association of shorter telomeres with increased risk for cancer. But that could be due to biases associated with sampling cases and controls at different times, or there might be some kind of reverse causality. And so look at the genetic versus observational study. So, by the way, those associations of telomere length with cancer risk were quite heterogeneous. So while there were some that showed an association of short telomeres with increased length, I think most of them were going the other way, with longer telomeres being bad. So if you look at sort of meta-analyses and versus this Mendelian randomization, you see that in this case they actually are fairly similar, with maybe the genetic Mendelian randomization estimate being a little bit worse. That longer telomeres are worse than what we got from meta-analysis. And actually, it turns out you can, because those estimations that I showed in the beginning for Mendelian randomization can be, so you can do the analysis of the genetic factors on the phenotype in one population and the genetic factors on the disease outcome in the other population, and then you can just put the two estimators together, and so you can quickly scan through lots of possible associations between diseases and certain intermediate phenotypes, or you can do lots of intermediate phenotypes in a specific disease. And in this slide, what's being done is comparing telomere length versus lots of different diseases, and lung cancer turns out to be the most significantly associated outcome for telomere length, and so it's definitely bad. It's also bad for lots of other cancer, especially bad for glioma. And on the other hand, for some lung diseases like interstitial lung disease or idiopathic pulmonary fibrosis, having short telomeres is a risk factor. I think the take-home message from this slide is, despite what you might see on TV, don't try to make your telomeres longer, because while it might protect you from cardiovascular disease, it may increase your risk for cancer quite dramatically. So this is, like I was saying earlier, you can apply the same kind of approach to lots of different phenotypes for a specific disease. And when you do that for lung cancer, you find, as you should, that smoking is the most important factor, but also turns out vitamin B12 is also a risk factor. So having high vitamin B12 is bad. We don't really know why. This preliminary result has been confirmed now in a paper that I think just is coming out in JAMA Oncology. So I'm going to close this pretty soon. There's a pretty complicated study that was recently published doing the same kind of analysis with the metabolites. So they did a metabolome study, and they looked at lots of SNPs and connected that to lung cancer. When they do that, they find arachidonic acid metabolite being very, very strongly associated with lung cancer risk. So that's sort of allowing us to screen through lots of new sort of phenotypes that might be associated with lung cancer. There are some older studies. So bilirubin has been associated with protection from lung cancer, and there's, for example, surfactant protein B is also a fairly robust biomarker for the development of lung cancer. I did want to just mention this truly massive study that was done. It was really designed to look, I think, for cardiovascular disease in bilirubin, but they studied about 500,000 people. And what they found, so these are people with normal ranges of bilirubin, but it just kind of varies. There is Barre Syndrome as an example where people are born with a mutation in UGT1A1 that influences their ability to metabolize bilirubin, and so when they're when they're very young, they have to be sort of treated for that. But anyway, there's lots of variability in the population. It turns out that that higher bilirubin is protective from lung cancer and COPD, and also people with Barre Syndrome are protected actually from pulmonary diseases. So those things go together. So I just want to thank all the people I collaborate with. This is only a small subset of them, but we work very well in teams. We have mechanisms for sharing our data, and we always welcome that within the constraints that we try to make the analyses as consistent among groups so there's not too much overlap. And so summarizing, there are high-risk mutations in retinoblastoma, P53, some very rare mutations in EGFR, and then we have a region on 6q that is influencing familial lung cancer. So these are uncommon, but they can shed quite a lot of insight into the etiology of lung cancer. There are nicotinic receptors, region on chromosome 15, and there's a couple other nicotinic receptors now that are implicated. Also, the HTERT region is influencing lung cancer risk, and those are in common across the different cancers. But if you look at adenocarcinoma and squamous carcinoma, they look almost completely distinct, as if they're really not related in terms of their basic etiology. So I didn't talk about this too much, but the squamous carcinomas have often mutations in homologous recombination repair, including mutations CA2, RAD52, CHEK2, and HMSH5, and also the HLA region. The DNA repair genes seem to be more relevant for adenocarcinoma. We found that long telomere length increases lung cancer risk, and BMI also appears to contribute, especially to squamous and small cell carcinoma. Genetic information is providing a great number of new insights into the etiology of lung cancer and other common cancers, and seems to be particularly useful for identifying host factors that have a genetic influence. Usually, when we do Mendelian randomization, it's quite informative, but we also like to follow that up with a more direct analysis of trying to show that the factor studying is directly related to the disease outcome. So we would typically, do Mendelian randomization as a way of scanning through markers and then do additional studies. But I think this method is turning out to be quite informative. As we go forward in time, a combination of genetic, epidemiological, and biomarker panels will be needed for risk stratification. And one other comment here is that unfortunately most of our studies have been done on European descent. There are studies in Asians and Asian descent, and truly underrepresented at this time are populations of African Americans and Hispanics, and that partly reflects the difficulty in collecting sample populations. They tend to be less common at every site, so in order to do the studies, you have to get multiple investigators to participate, and also in some cases reluctance from the affected individuals to participate in some studies. We need to make efforts to improve the participation in other populations. African descent populations are of particular interest for lung cancer, prostate cancer, and some other cancers that are more common in those groups. And they're also useful for understanding the genetics because they have a more diverse genetic background. They are many different groups, and so they are more informative. It's just been a limitation of a lot of these studies up till now. So thank you very much for listening, and I'm happy to take comments. I'm a little sorry that I only talk about lung cancer, but it's something that I am very interested in studying. So thanks a lot.
Thank you, Dr. Amos, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window, type your question into the box that appears on the screen, and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Dr. Amos, our first question is, what kind of design would be best for identifying a common variant with a low level of effect on disease risk?
Well, for that kind of design, we would usually use a case control design. And the reason we do that is because it's fairly easy to collect cases with the disease and controls without the disease. And then the costs of the analysis are also not so high these days because we can usually use an array-based technology to identify those genotypes that are associated with the case status. And then we have known linkage disequilibrium patterns. Those are associations among alleles that allow us to infer most of the variability across the entire genome.
Our next question is, why could testing for the effects of BMI on lung cancer risk lead to an erroneous finding if incident lung cancer cases and controls were studied?
So again, I explained the reason for that is because cases may have been sick before they came to attention and lost weight because of that, or if they smoke a lot, then they tend to be thinner.
And it looks like we have time for one more question. Why are consortia needed to identify high-risk variants influencing lethal diseases, and how can we use family data to overcome limitations in accessing enough disease-affected individuals?
So we need consortia because those families that are informative are harder to find, and so you need to be able to share the samples and the data among the different places that can collect that kind of information, and infer the genotypes from the offspring and the individuals who married into the family, so that those will allow us to infer the genotypes of any individuals who were diseased and also died before you could collect samples from them.
I would like to once again thank Dr. Amos for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.